I'd like to thank everybody for attending this talk. I'd like to thank the organizers at BioConference Live for inviting me to make the presentation. I'm going to talk to you today about the post-translational control of the molecular chaperone HSP90, but before I get to that particular topic, I want to put HSP90 in context and explain why it's an important novel molecular target in cancer. So as you can see from this title slide, HSP90 as a molecular chaperone has roles to play in the function of normal cells, but it's hijacked by cancer cells. HSP90 is ubiquitously expressed in all kingdoms except for archaea. In eukaryotes, there are a number of orthologs of HSP90. In yeast, which is a very useful model to study HSP90 structure and function, there are two genes, and a stress-inducible HSP82 and a constitutively expressed HSC82. These genes are highly homologous, although not completely identical. Deletion of both is lethal, so yeast cells need at least one HSP90 gene to be expressed for viability. In humans, there's a similar situation. There's a beta isoform that is constitutively expressed. Its knockout actually is embryologically lethal. And there's an alpha isoform that is stress-inducible. Mice can survive knockout of HSP90 alpha, but males are sterile. Humans also express two other HSP90 related genes. One, called GRP94, is a molecular chaperone that's restricted to the endoplasmic reticulum, and another, called TRAP1, is restricted to the mitochondria. So the obvious question, why target a housekeeping protein like HSP90 in cancer? As I said, in normal cells, HSP90 is part of the proteostasis network that helps to maintain homeostasis. Cancer cells have utilized the proteostasis network of proteins, including HSP90, to maintain the function and stability of mutated and overexpressed genes, many of which are oncogenes. Another way of looking at this is that a normal function of HSP90, both at the cell, tissue, and organismal level, is to sense environmental stress and to coordinate a cells and tissues and organisms' responses to such environmental stress to promote survival. So it's not a big leap to consider the fact that cancer cells generally exist at all times in the presence of one type of environmental stress or another. As diagrammed here, HSP90 is part of a circuit that reads nutrient stress, stress from the proteome due to misfolding, hypoxic stress, and the genetic instability and aneuploidy for which cancer cells are well known, and turns on and regulates numerous pathways in the cell that helps the cell, in this case the cancer cell, cope with these different kinds of stresses. On the far right of the slide are just a listing of a few of the client proteins that depend on HSP90 for their function and stability. And these proteins help to modulate the pathways that cancer cells use to protect themselves from environmental and chemical stress. This model is based on a crystal structure of bacterial HSP90. Bacterial HSP90 and mammalian HSP90 are actually not that very different. The domain structure is maintained. As you can see, there's an endodomain to which ATP binds, a middle domain, which is a site for co-chaperone and substrate binding, and a C-terminal domain, which serves as a dimerization site. This is a model of one protomer of HSP90, but HSP90 exists as a dimer that is dimerized through motifs in the C-terminal domain of each protomer. ATP binding and ATP hydrolysis are essential to the function of HSP90 in vivo. This is a simple model showing that in the presence of ATP, which binds to the endomains of the protomers come together to dimerize in a transient fashion, and this allows HSP90 to hydrolyze the ATP, and this binding, hydrolysis, and conformational dynamics are all essential to the ability of HSP90 to chaperone its clients. Now, the story is more complicated than that. HSP90 does not work on its own. Its ATPase activity in the absence of accessory proteins is extremely low, and a number of accessory proteins termed co-chaperones, and shown here, help to control the rate of ATP binding and hydrolysis. The two co-chaperones shown in red slow down the chaperone cycle and the conformational dynamics upon ATP binding, and this is important for loading client proteins onto HSP90, whereas AHA1, shown in green, helps to enhance the ATP hydrolyzing activity of HSP90, and I'll talk more on this later. So this is a close-up of the endomain of one protomer of HSP90, on the left showing where ATP binds in a unique pocket in the endodomains of each of the protomers. On the right are two of the original HSP90 inhibitors that were identified in the early and middle 1990s, galdanamycin and radicicol. And the point of this slide is to show that these inhibitors bind to the ATP pocket and replace ATP in that pocket. What this slide shows is a clinical development of HSP90 inhibitors over time. So galdanamycin was identified in 1970. We identified HSP90 as its molecular target in 1994. The first HSP90 inhibitor to enter the clinic, a derivative of galdanamycin called 17-AAG, began clinical trial in 1999. And since then, 18 chemically distinct HSP90 inhibitors have entered the clinic. Until 2006, these HSP90 inhibitors were all versions of galdanamycin. But in 2006, the first totally synthetic HSP90 inhibitor was identified and tested. And since then, a number of synthetic HSP90 inhibitors have reached various stages of clinical evaluation. To date, one HSP90 inhibitor, STA9090, also known as gantenerumab, is in phase 3 clinical trial. So to go back to the model of the HSP90 chaperone cycle, which involves a number of conformational changes together with ATP binding and hydrolysis. The role of drugs in blocking the HSP90 cycle occurs because these drugs displace ATP, thus preventing the conformational changes that ensue upon ATP binding and which are essential for HSP90 to chaperone its clients. So in the presence of these drugs, such as galvanomycin, radicicol, and all of the synthetic inhibitors that have been developed since, they block the chaperone cycle, and client proteins are delivered to E3 ubiquitin ligases that cause their ubiquitination and then subsequent degradation by the proteasome. So client proteins of HSP90 in the presence of HSP90 inhibitors are ubiquitinated and generally degraded in the proteasome. Two E3s have been identified that promote this process, one called CHIP and another called Cullen-5. There are likely more E3s as well. This is an example of what happens to a typical HSP90 client protein, the kinase BRAF, in the presence of an HSP90 inhibitor, in this case 17-AAG. This is a Western blot showing that over time, the level of the client declines to levels that are almost undetectable within 16 to 24 hours of drug treatment. In some cases, depending on the sensitivity of the client, this decay in protein expression and stability can be even faster. And it's important to note that this is not an effect on mRNA, it's not an effect on transcription, it's an effect on protein stability. And this loss of BRAF protein is secondary to its ubiquitination and degradation in the proteasome. Not all proteins, of course, are clients of HSP90, and sometimes proteins that are very closely related may diverge in their dependence on HSP90. And we show that here with the kinases ERB2 and ERB1, ERB1 also known as EGF receptor, much less sensitive to HSP90 inhibition, whereas ERB2 is exquisitely sensitive, showing here within 4 to 6 hours, ERB2 protein is lost from tumor cells, whereas it takes much longer, 16 to 24 hours, to have an effect on ERB1 expression. Again, even though the loss of ERB2 is so quick, it really depends on its ubiquitination, as shown in this slide from a paper we published in 1996. Within 15 minutes of drug treatment, the levels of ERB2 protein shown on the left begin to decline, but this is accompanied by dramatically enhanced ubiquitination. If one were to include a proteasome inhibitor in this experiment, you would see that you would protect the client protein from degradation, although in many cases the client will become insoluble and precipitate out of the soluble fraction of the cell. Another client that is very dependent on HSP90 and thus very sensitive to HSP90 inhibition is the kinase EML4-ALK, which is characteristic of certain forms of lung cancer. As you can see in the Western blot on the lower left, EML4-ALK, both the phosphorylated and thus active form, as well as the total ALK, is even more sensitive in this experiment than HER2 or ERB2. And in an animal experiment, one can see regression of tumors that are driven by EML4 ALK when animals are treated with an HSP90 inhibitor. So one way to think about HSP90 inhibition as an alternative strategy to inhibit a number of kinases in the cell with a single stroke, as opposed to using TKIs, which have specificity for a particular type of kinase or family. HSP90 inhibition is able to target a number of kinases that are HSP90 dependent at the same time. So to turn to some clinical evaluations of HSP90 inhibitors in cancer patients, a study published a few years ago was the first to demonstrate the efficacy of an HSP90 inhibitor, and this was in HER2 positive, ERB2 positive breast cancer patients that had been treated and responded to Herceptin, but eventually progressed on Herceptin alone. The researchers showed that when HSP90 inhibitor, in this case 17-AAG, was added onto Herceptin, partial responses were restored, and these are all patients that were progressing on Herceptin alone. The response rate was 26%, and the overall rate of benefit was 63%. This has now been validated for other HSP90 inhibitors. Phase 2 analysis of non-small cell lung cancer has shown that the HSP90 inhibitor gantenerumab as monotherapy has significant activity in ALK-positive lung cancer. So these are lung cancers that are driven by the activated form of ALK that I was discussing in the previous couple of slides. And here, 75% of the patients examined in this trial that were ALK-positive responded to gantenerumab as a monotherapy. This is an example of a patient's tumor. This patient was on crizotinib, the ALK inhibitor, for 1 year before progressing. When this patient was then administered 3 doses of gantenerumab, the HSP90 inhibitor, you can see that there was additional shrinkage of the tumor that was no longer responsive to the ALK inhibitor. So it's clear that tumors that are driven by highly sensitive HSP90 clients, such as HER2 or ERB2 and ALK, are most likely to be responsive to HSP90 inhibitor therapy in vivo. This is an example of a non-small cell lung cancer patient whose tumor is BRAF dependent. As you remember, BRAF is also an HSP90 client. This patient had been on a number of therapies. The average duration of each was from 2 to 4 months. After lacking responses over the course of 13 months to the various drugs shown in the slide, after 4 cycles of gantenerumab as monotherapy, there was clear evidence of tumor shrinkage. This is a patient with triple-negative breast cancer, so not dependent on HER2, and actually it's not clear what drives this particular form of breast cancer. and yet it also responds to single agent HSP90 inhibitor after not responding to a series of therapies. So HSP90 inhibition is likely to have a broad applicability in a number of different types of tumors that are driven by different molecular targets. Is there evidence that the amount of HSP90 that a tumor expresses is predictive of patient survival? And the answer to that is yes. The initial study showing this was published a few years ago, looking at a series of non-small cell lung cancers prospectively. Non-small cell lung cancer is genetically very unstable, and so there are many gains and losses in gene expression. So the green here are gains in gene expression, multi-copy genes. The red are deletions of various genes and portions of chromosomes. So if you look closely, you can see that the HSP90 alpha locus is among the regions that are not uncommonly deleted in non-small cell lung cancer. So what this study did was to look at a series of 32 patients with non-small cell lung cancer and to look at their overall survival and correlate that to the level of HSP90 expression or to the deletion of the portion of the chromosome that encode the HSP90 alpha gene. So in panel A, if you look at HSP90 expression high or normal, you can see that normal expression of HSP90 compared to low expression of HSP90 had markedly different overall survival. Low expression of HSP90 predicted for better overall survival, and this was validated in 2 additional sets of tumors. So in non-small cell lung cancer, low expression of HSP90 predicts for better overall survival. More recently, this was also shown to be the case in breast cancer in another paper, where the investigators did a similar analysis, in this case looking at recurrence-free survival of triple-negative breast cancer, and finding that those patients whose cancers had upregulated HSP90 expression had a shorter recurrence-free survival time than did everybody else. So again, the expression level of HSP90 is a negative prognostic marker both for overall survival and for recurrence-free survival, suggesting that targeting HSP90 is going to inhibit something that drives cancers. And there is clinical evidence now that this is the case for triple-negative breast cancer, for non-small cell lung cancer, and for HER2-positive breast cancer. So for the second half of the talk now, I want to focus on the regulation of HSP90 in normal cells, but also in cancer cells. And this regulation is by post-translational modifications to HSP90. And by this term, I mean phosphorylation, acetylation, ubiquitination, etc. All of the modifications shown on this slide have been identified by 90, and many of them have been shown to affect not only drug sensitivity, but also HSP90 activity itself, its location inside the cell, and I'm going to show you some examples of each of these. focusing on phosphorylation as the post-translational change because phosphorylation has been the most actively investigated of the modifications to HSP90. So this is a slide showing HSP90 alpha and beta, the stress-inducible form and the constitutive form, listing some, but not all, of the phosphorylation sites that have been identified. As you can see, many of the sites occur in both HSP90 alpha and beta since the proteins are highly homologous. But there are two sites in particular outlined in green that occur only in HSP90 alpha and not in beta. These are threonines 5 and 7 in HSP90 alpha. And so I'm going to give you 3 examples of how phosphorylation or the lack thereof of either threonine 5 and 7, Y38 in HSP90 alpha, and Y313 in HSP90 alpha affect drug sensitivity, HSP90 activity, and location inside the cell. For threonines T5, for threonines 5 and 7, we know the kinase that phosphorylates these residues. It's DNA PK, which is itself a client of HSP90. For Y38, we know the kinase is WEE1, also a client of HSP90. And for Y313, we know that, yes, a closely related protein to C-SARC is able to phosphorylate this residue, although we also know that it is not the only enzyme that is able to do so. Yes is also a client of HSP90, as is SARC. So a theme that's appearing here is that many of the kinases that are clients of HSP90 seem also to be able to phosphorylate the protein with different impacts on its location and its function, as well as its drug sensitivity. So first, let's look at this residue in the end domain, which is Y24 in yeast and 38 in mammalian HSP90 alpha, that is phosphorylated by WEE1. Pharmacologic inhibition or silencing of WEE1 sensitizes both yeast and cancer cells to HSP90 inhibition. And what we're showing here is we're looking at the percent of apoptosis and escalating doses of the HSP90 inhibitor 17-AAG. You can see that on its own, at these concentrations, 17-AAG does not induce a high degree of apoptosis. But if we include a WEE1 inhibitor at a dose that by itself is minimally apoptotic, the combination of HSP90 inhibitor and WEE1 inhibitor induces synergistic apoptosis. This is true in 2 different tumor lines, PC3, which is derived from a prostate cancer, and HeLa cells, which is derived from cervical cancer. So combination of WEE1 inhibitor and HSP90 inhibitor is synergistically apoptotic. This is also true for the newer synthetic HSP90 inhibitors. We're showing in PC3 cells here that 2 of these drugs, STA9090 or gantenerumab and SNX2112, also demonstrate synergistic apoptosis when combined with low-dose WEE1 inhibitor. That in itself is not apoptotic. These results can be translated to animal studies looking at the growth of tumors. Here we're using a model of PC3 in xenograft form. We're using doses of 17-AG and WEE1 inhibitor that are low enough so that on their own as single agents do not cause significant inhibition in tumor growth. But when combined at these doses, which you can see in the orange line, show significant growth inhibition. Again, demonstrating synergy between very low dose WEE1 inhibitor and HSP90 inhibitor. And since WEE1 inhibitor is currently undergoing early clinical evaluation, it should be possible sometime soon to perform a clinical study in cancer patients combining WEE1 inhibitor and HSP90 inhibitor. The second example I want to talk to you about is how phosphorylation can affect HSP90 activity. So as I mentioned, HSP90 has very low ATPase activity inherently. And in order for it to be functional in cells, its ATPase activity has to be increased. This is performed by a co-chaperone, an accessory protein called AHA1, which stimulates the inherently low ATPase activity of HSP90, as shown on this slide, up to 10-fold. But in cells, there is much more HSP90 than AHA1. So mechanisms must exist for HSP90 populations in cells to recruit AHA1 in order to stimulate ATPase activity. This is just showing a cartoon which I showed you a number of slides ago, showing that the chaperone cycle is dependent on AHA1 to promote ATP hydrolysis. So here is the proximal location of Y313. It's in the M domain of HSP90. What we initially found was that if we made a phosphomimic mutation of the tyrosine, which is Y313E, shown on the right here, and immunoprecipitated either transfected wild-type HSP90, flag HSP90, or transfected flag HSP90 Y313E, and you can see we're immunoprecipitating equivalent amounts of HSP90. Endogenous AHA1 associates much more profoundly and abundantly with HSP90 Y313E compared to wild-type HSP90. These experiments were done in 293 cells. If we look at the purified proteins AHA1 and HSP90 and measure affinity of AHA1 for wild-type HSP90 compared to HSP90 Y313E, you can see that even with purified protein measurements, there's nearly a four-fold increase in the affinity of AHA1 for the phosphomimic HSP90 Y313E compared to wild-type HSP90, consistent with the pull-down data obtained in 293 cells. We then went on to make an antibody specifically recognizing phospho Y313 HSP90 and used it in an experiment where we immunoprecipitated total HSP90 with an antibody to HSP90 called AC88 compared to an immunoprecipitation of AHA1. And for this purpose, we transfected 293 cells with flag AHA1. We then Western blotted for HSP90 in the upper panels, and Western blotted for phospho Y313 HSP90 in the lower panels. And what you can see is that the HSP90 that is co-precipitating with flag AHA1 is much more likely to be phosphorylated on Y313 than is the general population of HSP90. This is graphically shown underneath the Western blot, where we've made a ratio of HSP90 phospho Y313 over total HSP90. And from this graph, you can see that the HSP90 associating with AHA1 is 7-fold more phosphorylated on Y313 than is the general population of HSP90. All these data taken together suggest that phosphorylation of Y313 may serve as a recruitment signal in the context of the cell for a pool of HSP90 to recruit AHA1, and therefore this might be predicted to be an actively chaperoning population of HSP90. To confirm that AHA1 is better able to increase the ATPase activity of phospho Y313 HSP90, we go back to purified protein data here showing that AHA1 is more potently able to stimulate the ATPase activity of equal amounts of Y313E HSP90 compared to wild-type HSP90, demonstrating again that phospho Y313 HSP90 is likely to be much more readily activated in terms of its ATPase activity as a consequence of recruiting AHA1 to the complex of chaperones. So I mentioned a minute ago that this may also identify actively chaperoning pools of HSP90, and we did an immunocytochemistry experiment to see if we could see distinct populations of HSP90 in the cell using the antibody to phospho Y313. This is a stain looking at total HSP90 using an antibody that generally recognizes HSP90 in the cell. The green signal is HSP90, the blue signal is a nuclear stain. So what this shows is what is readily accepted, that nearly all of HSP90, roughly 95 to 97%, is cytoplasmic. There is a small amount in the nucleus, but the vast majority of HSP90 is cytoplasmic, but it's relatively uniformly distributed in the cytosol. In contrast, if we look to the pattern of staining we see with the antibody to phospho Y313 HSP90, you see that in these two bladder cancer lines, the pattern is distinctly different from total HSP90. In RT4 cells shown on the left, there's a punctate pattern of distribution. In TCC cells shown on the right, the pattern is even more distinct, where it appears that phospho Y313 HSP90 is localized to the periphery of the cytoplasm, perhaps on the inner portion of the membranes of these cells. So the question remains, are these sites of actively chaperoning HSP90? And this is an area of intense investigation at present. But it is clear from these experiments that unique phosphorylation of HSP90 is able to identify unique pools of the chaperone in the cell. Another example of this is going to be shown here, looking at phosphorylation of threonines 5 and 7, in this case only in HSP90 alpha, since these residues are not threonines in the beta isoform of HSP90. Before getting to that, I want to introduce the extrinsic apoptotic signal pathway that cells use. This refers to death receptors that exist on the plasma membranes of cells that are activated by ligands binding to them, which in turn activates a caspase cascade leading to nuclear apoptosis, as evidenced by DNA fragmentation. Trail, a peptide, is one of the ligands of these death receptors, as shown in this cartoon. So trail binding to an activation of these death receptor pathways leads eventually to nuclear fragmentation and nuclear apoptosis. This pathway is distinct from the pathway of intrinsic apoptosis, which I'm not going to discuss here. Very shortly after trail binding to cells, within minutes to a few hours, gamma H2AX, which is a phosphorylated form of histone 2AX, appears in cells that are destined to undergo nuclear fragmentation, and is shown by the red stain in this picture. These experiments are done in HCT116, which is a colon cancer line. So in this particular picture, although there are a number of cells, as evidenced by DAPI staining, which is a nuclear stain in the field, 2 of the cells are currently positive for gamma H2AX, and so are beginning to undergo nuclear fragmentation. On the right, in the green color, you see that the same cells that are positive for gamma H2AX are also positive for phosphorylated threonines 5,7 HSP90 alpha. And if you overlay the picture, we can confirm that the same cells positive for phosphorylated H2AX are also positive for phosphorylated T57 HSP90. So if you remember, I mentioned a minute ago that 95 to 97% of HSP90 is cytosolic. Perhaps 3 to 5% is nuclear. This phosphorylation of threonines 5 and 7 identifies a nuclear population of HSP90 that's phosphorylated in response to this extrinsic apoptosis-inducing agent. One can see that this is DNA PK dependent. So in panel A, we use inhibitors of ATM kinase that's also involved in trail activated apoptosis, and an inhibitor of DNA PK, and only the inhibitor of DNA PK is able to inhibit phosphorylation of threonines 5 and 7 in HSP90 alpha in response to trail. In panel B, we're looking at cells where DNA PK is knocked out compared to parental cells, and in the cells where DNA PK is knocked out, there is absolutely no phosphorylation of threonines 5 and 7 in HSP90 alpha in response to trail. Importantly, there is also no phosphorylation of gamma H2AX in these cells, demonstrating that in HCT116 cells, trail-induced phosphorylation of both H2AX and threonine 5,7 HSP90 alpha is DNA PK dependent. Here, to make things a little more complicated, we're showing that DNA PK activation by trail actually requires HSP90 alpha. If you remember, I mentioned a short while ago that DNA PK is a client of HSP90, and so HSP90 is required to maintain the kinase in a metastable state that is able to respond to trail-induced activation. And here we're showing, in fact, that in cells where HSP90 alpha is knocked down with siRNA, in panel A, there is minimal appearance of phospho T57 HSP90 in response to trail. There's minimal appearance of gamma H2AX in response to trail. And this is in the presence of the beta isoform of HSP90. So this phenomenon of T57 phosphorylation and gamma H2AX phosphorylation by DNA PK is HSP90 alpha dependent. This is a unique role for HSP90 alpha in the nucleus of cells. In panel C, we're just quantifying the reduced amount of gamma H2AX and T57 phosphorylation when HSP90 alpha is knocked down. And in panels D and E, we're looking at measures of apoptosis. And in the gray bars, which reflect apoptosis in cells knocked down for HSP90 alpha, you can see reduced apoptosis, reduced nuclear apoptosis, compared to the black bars, where wild-type HSP90 alpha is normally expressed. So, in sum, these data demonstrate that HSP90 is regulated both functionally in terms of drug sensitivity and in terms of location in the cell by post-translational changes. We've given you several examples here where phosphorylation of different residues in HSP90 can have these different effects. And in some cases where antibodies exist to the phosphorylated residues, it's been possible to document, in addition to making phosphomimetic or non-phosphorylatable HSP90 mutants, that these modifications actually occur in cells, either under steady-state conditions or when cells are challenged by environmental stress. So, taken together, I want to leave you with the understanding that HSP90, an important part of the proteostasis network of normal cells, is hijacked by cancer cells to promote survival in the face of genetic instability, chemical stress, and environmental stress. And that one has to keep in mind that there's an interplay between HSP90 accessory proteins termed co-chaperones and post-translational changes, all of which work together to allow normal function of the chaperone machinery. This requires ATP binding and the resultant conformational changes that are set in motion. And HSP90 inhibitors, by displacing ATP from HSP90, prevent these conformational changes, collapse the chaperone machinery, and prevent HSP90 from chaperoning its clients, more so in cancer cells than in normal cells. And with that, I would like to acknowledge the folks in my lab over the years that have performed all of the experiments that I've shown you in this talk, and I also would like to acknowledge our collaborators from the medical oncology branch and laboratory and molecular pharmacology at NCI, and also our collaborators at the University of Sussex in the United Kingdom. With that, I would like to thank you for your attention. I hope you enjoyed the presentation and found it informative. And would just like to add that you should go to this website, bioconferencelive.com/CME-CE to obtain credits for continuing education. So that's it. Thanks for your attention, and goodbye.